This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 757, recorded on May 18th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here, it's a beautiful blue sky with some wispy clouds, 76 Fahrenheit, and I haven't converted it to Celsius yet. Sorry.
Here it's 26 Celsius. Really nice day out. It's warmer out than it is in here. It's always cold in here. Also joining us from Austin, Texas, Rich Condit.
Hi there. We got 78 degrees, cloudy. It rained like crazy yesterday, and it's supposed to rain like crazy again starting tonight and all day tomorrow, to the point where we are under a flood watch, flash flood watch. So this is very cool, and it ought to help bring the reservoirs up and bring us up to normal rainfall. It's all good.
Near a creek or a river or something?
No, we're okay.
Okay.
And from Madison, New Jersey, Brian Barker.
Hi, everybody. I am sort of shocked right now because apparently I am winning the temperature sweepstakes. It's 81 Fahrenheit, 27 Celsius here, and beautiful blue skies. It's a great day.
So we are on 757. We're going through the Boeing aircraft numbers.
Thought about that, too.
Daniel wanted 747. He got it.
Upper deck. I forget whether the 757 has some sort of special name.
I don't know. It's just a six-seat thing. It's nothing. It's not even a wide body. It's just a long guy.
Aluminum tube.
Aluminum tube, yeah. Crazy stuff that we do. Okay. Before we get started, don't forget to check out a vaccine education town hall, asv.org/education. Is that enough, or you want to say more, guys?
Tell your friends. I did one last night. We had a good time. Good questions. Has
the most popular question changed recently?
Which was the most popular question?
I don't know. Just whatever they always.
I had to ask the question about the CDC lifting its mask requirements myself. I expected people to be, climbing all over that, and no, that wasn't happening. We are at the point where those who are vaccine hesitant are really important to reach.
Yeah.
Okay. Because we have, there's, in terms of logistics, from what I can see, very little reason not to be able to get a vaccine. I'm sure that there are places where it's logistically more difficult. Yes. Okay. And I don't want to downplay that, but vaccine is available, put it that way. And it's also clear that, on a population scale and an individual scale, they're enormously effective. And the way to beat this thing is to get people vaccinated. There's no downside. So if you have questions, understandable, give us a ring. Sign up for a town hall. Ask our questions.
Daniel said last week that 99% of the patients coming into hospital are not vaccinated.
Sure.
Right. Anywhere you hear of this person or that person infected, they're probably not vaccinated.
I looked up. CDC has there's the CDC has a lengthy description of how it came to its decision to basically lift the mask restrictions for vaccinated individuals, and it has another page that I was turned to about vaccine breakthrough, okay, where they summarize what has gone on. And now this is, as I recall it, it's like self-reported, kind of, sort of, okay, or reported by cases that show up. And so there are those who argue that it's an incomplete data set. because it's not like actively going out and gathering these numbers. Okay. Not only that, but they were tracking any confirmed SARS-CoV-2 infection in a vaccinated individual, and that became cumbersome and probably inaccurate. So now they're just doing hospitalizations and deaths. And if you look at that, out of 115 million vaccinations in the U.S., there have been on the order of 1,300 cases of COVID and 280-something deaths, okay? So that's about 1 in 90,000 vaccinated people hospitalized with COVID, and 1 in about 500,000 deaths. Most of those were in people over 65 years old, and they didn't break it down into, comorbidities and that kind of stuff that I could see. But yeah, I mean, there is some breakthrough, but it's very rare.
Yeah. And we need to remind people that, breakthrough in terms of disease is the problem that we worry a lot about. Breakthrough in terms of infection is something that we thought might happen, that we might have a little bit of viral replication in some people. And that probably happens with some other vaccines.
Most likely, sure.
And
absolutely.
We're just not testing. You don't get tested for measles weekly after you get your measles vaccine to know if you have any breakthrough viral replication. So, hearing stories about infection in vaccinated people is not necessarily a big surprise.
Right. It's really weird because I'm feeling, at least intellectually, pretty bulletproof. Okay, being fully vaccinated, but after hunkering down for a whole year, it's a little creepy to expose yourself to the rest of the world, but it's justified.
Yeah.
Well, the numbers are saying, yeah, you're pretty good in terms of serious disease. Yeah.
Yep.
But remember, as Brienne said, this is nothing new. You get infected after vaccination. You just don't get sick. You probably don't transmit. Only because of the big focus on this pandemic is this called breakthrough infections. But we don't call that with polio when you get infected after you're vaccinated or anything else, because there's not similar emphasis. But it's not a big deal, as I tell people all the time. It'll be fine.
Rich, what was the number that you quoted? Because I have a 1 in 10,000 vaccinated having breakthrough cases from numbers I found on the CDC site.
Let me see here. Hang on. Let me get just give me a second here, because I saved the link. Okay.
Yours was based on 150 million. Mine was based on...
No, this is 115 million.
115. Okay, yeah.
115 million. And if we consider just the hospitalizations and death, this is as of May 10th, 2021, on the CDC site, out of 115 million people in the United States fully vaccinated, there were 1,136 hospitalizations. So a total of, if you include hospitalizations and deaths, a total of 1,359 cases that resulted in either hospitalization or death.
Okay.
And of those, 223, I had the numbers wrong. 223 were deaths. So if you take 115 million divided by, let's do the deaths, 223, that's 1 in 500,000. Okay.
Right. Okay, because so mine was slightly before that, the numbers for the CDC, and it was 95 million and a total of 9,245 cases, which comes out to 1 in 10,000 cases.
Right. Okay.
And they say that could be an undercount, of course, because they don't catch all of the asymptomatic cases. But
right.
This was because I was working out an answer for someone who was worried about the 95% efficacy. They wanted to know, does that mean I have a 5% chance of getting infected? It's like, no, it's like 1 in 10,000. vaccinated people get infected.
Right, and your number is higher because they're counting people who were infected but not hospitalized.
Exactly.
And that just became unwieldy,
Right.
According to this that I've been reading.
Okay.
There was Daniel cited a study in MMWR. Yeah, it was among healthcare workers. Interim estimate of mRNA vaccine effectiveness preventing disease among healthcare workers. Millions and just a few thousand actual disease. I can't remember the citation for that, but that if you're worried about not a careful study, as you said, Rich, that one was carefully done because it was in healthcare workers that they followed regularly. All right. Now we have a follow-up from our last episode where we talked about the new OPV2, one of the co-authors, Andrew McAdam, has written. What a nice surprise to hear you all talking about NOPV2 on TWiV. Wonderful discussion. Thanks for your kind remarks about our paper and for your supportive review. The paper eventually came out in May 2020. As you rightly say, the concepts behind the vaccine design came out of basic scientific research in many labs, arguably starting with infectious cDNA and the analysis of David Miner's nappies.
I wonder if people know that story.
No, they don't.
We've told it before.
David Miner was the son, or still is the son of Phil Miner, a British poliovirologist, and David was he and his wife's first kid. And the story goes, this is now early 80s. They're talking about how do we use sequencing to study the polio vaccine. And Morag Ferguson, who worked in the lab, said, "Why don't you get your son's nappies and bring them in, and we'll sequence it." So that's the British term for diapers, I guess. And so David got his OPV, and then they took his nappies every day, and after, he's excreting OPV, and they sequenced it. And that's how they found within 36 hours, the type 3 component reverts in the 5' UTR. That was the first indication that something, at the molecular level, we had already known from the early trials in the 60s that when you fed OPV the people, what they excreted, if you put that back into monkeys, it would paralyze them. We already knew something was going on because if you take the vaccine and put it in monkeys, it doesn't paralyze them. If things go well, this could be a great example to use in future grant proposals. Bench to bedside. It's bedside. To update you with what's going on in case you're interested, results of the phase 2 trial and a second phase 1 are now published in Lancet, and it provides the links showing very similar findings to the first phase 1, but in more and younger people. We all know polio can usually find a way to revert given enough opportunity, but the hope is that the frequency of this will be reduced sufficiently to prevent new circulating vaccine-derived polioviruses. Remember that monovalent OPV2 gives rise to circulating vaccine-derived poliovirus transmission rarely, and even a tenfold reduction in this would have a big impact. Of course, we anticipate even bigger reductions. That's a good point. The reversion is quite rare. Well, circulating is rare. The reversion happens in everybody. So maybe you don't need to cut back much. Well, we'll see. Other clinical trials are in progress or are planned, including a phase 3 required for full licensure, a trial in naive infants, and phase 1 studies of NOPV1 and NOPV3. So you may ask, why do we need NOPV1 and 3? Well, so those two 3 has been eradicated, right? But if it came back, you would want to stop the outbreak with OPV, and it would be good to have NOPV instead of putting back all the revertants, right? And 1, of course, is still ongoing, and maybe we could start using NOPV1. If I was to be pedantic, well, why not? Everybody else is, right? I would pick you up on a couple of things. Regarding recombination, I think you misremembered the observations in trivalent OPV recipients where intertypic polio recombinants are common. Recombination with other species C. antero has only been found in circulating VDPV isolates, to my knowledge. Yeah, that's correct. And for domain 5, attenuation correlates with thermodynamic instability rather than the other way you said it. That's right. It is hard for S15 domain 5 to regain stability and hence revert because that would require more than one double mutation converting a UA into a CG base pair. Destabilizing would be easy. Yeah, I misspoke. Also, on behalf of our CDC colleagues, the codon de-optimization in candidate 2 came from work in Kara Burns and Olin Q's lab, not Eckhart's. This candidate was found to be marginally less immunogenic than candidate 1 in the phase 2 trial, hence the decision to proceed with candidate 1. The lower/shorter excretion that you commented on could, of course, be advantageous with respect to onward transmission, but less so with respect to herd immunity a la Albert Sabin. Remember, that's a different kind of herd immunity. We actually get infected with the shed virus. That's why he says a la Albert Sabin. I like the subtlety there, the nuance. Happy to fill you in further if you have any questions. So Andrew is in the Division of Virology, National Institute of Biological Standards and Control. I would say is roughly the FDA over there. Now, people are going to write and say, no, it's not. But come on, that's what they used to tell me.
Okay.
Phil Miner used to work there, and I knew him very well, and Phil Miner's retired. And Andrew came up through Phil Miner. You know, he trained him, and now he's there and working on. That's a very cool story. Lovely.
Yeah, excellent. Thank you very much. That's a good good cleanup of that discussion.
Yep. We need lots of cleaning up here. At least I do. I do
Especially
anyway.
after the nappies discussion.
Nappies is great. It's a great story. So we have 2 papers for you today, a snippet and a paper. And the first is a paper published in PNAS from a group at the University of Colorado Boulder. The first author is Qing Yang, and the last author is Sarah Sawyer. Sarah Sawyer has been on TWiV 193 and TWiVo number 5. And go look at the art for TWiVo 5. Beautiful pictures of straw-colored bats. Really pretty bats. Anyways, just 2% of SARS-CoV-2 positive individuals carry 90% of the virus circulating in communities. All right. This is very cool. This is going to bring up a lot of neat things. So this is a screening program, data from a screening program at the University of Colorado Boulder, August 27th to December 11th, fall academic semesters. Dorm residents were tested weekly, and they had sites where you could go and get tested. Saliva collection, and they asked you if you were symptomatic or not at the time of collection. And so that's one of the important parts of this study. And this paper, they take 72,500 saliva samples. They screen them by RT-PCR, 1,405 SARS-CoV-2 positives. And most of them from unique individuals, right? Because if you're positive, then you go somewhere else to the healthcare system for further testing. And they have a lovely figure. It's probably open access, I would guess.
I think it is.
Where they have the distribution of the cycle threshold values of these 1,405 individuals, which gives you a lovely, indication of the spread that you would see. So, it's a log normal distribution. The mean is 2 times 10 to the 7th. Well, they say virions, but I'm going to say virus particles per milliliter. And get this, the highest load in one person was 6 times 10 to the 12th virus particles per milliliter. I don't know, it's a particle. It's an RNA copy, right?
Yeah.
Yeah, it is.
Genomes, I think, is the way to think of it. But that's not even necessarily accurate.
It's RNA copies, right?
It's RNA.
So it's totally wrong to say virions, right?
It is. And I found that very curious when I got to some later parts of the figure.
Yeah, because
or later parts of the paper.
10 to the 12th, that's more than we get with polio in terms of
So
infectivity.
10 to the 12th, for those who aren't used to thinking in exponents, which took me a few years to figure out, that's a trillion, right?
Trillion, yeah. It is a trillion.
And I used to, when I taught flu lectures to the medical students, I used to estimate the volume of a sneeze, the fluid coming out of a sneeze, as about 100 microliters. Okay? Mm-hmm. So that would be 1/10 of a mil. So we're talking 100 billion genomes in a sneeze.
And
So
and people can
watch out.
People can relate to trillions of dollars. That's what the pandemic has cost many nations. But the lowest load was 8 RNA copies per milliliter. Isn't that amazing? From
Pretty big range.
8.
And one of the things that's important about this is that they have these values. Okay,
Yeah.
A lot of testing, most of the testing that gets done, you just find out plus or minus. Okay, one of the important things about this is that it's quantitative.
Right.
Yes.
And at least here they talk about, this being asymptomatic individuals.
Yeah.
So they're seeing
Yes.
these numbers of viral genomes in asymptomatic individuals, which is really important to understand.
Yeah. My understanding was that if you came in and you were asymptomatic, you didn't go through this channel. They sent you off to the infirmary.
Yeah,
Okay,
that's right.
So they have only the asymptomatic people in this study.
But when we think about things like transmission from asymptomatic individuals and the 600 million RNA copies in Rich's sneeze, that starts to help you understand how there can be some transmission from asymptomatic.
Asymptomatic or pre-symptomatic. Okay, an important point of this that we will discuss is that this is one time point for any of these people, and you don't know where in the course of their infection they are.
Yeah, I mean, would many of them eventually get to a very high point as this, who knows, right? So they wanted to know if this range of our RNA copies, how it resembles what you would find in symptomatic patients. Okay, so they looked into the literature. They looked at published data sets from hospitalized patients. And of course, if you're hospitalized, then you're most likely symptomatic. Stay away from a hospital if you're not symptomatic, okay? Not that it's a bad place, but you shouldn't be there unless you work there. And they looked for saliva assays and where they had cycle threshold values. And so similar distributions, right? The mean 2 × 10⁷ RNA copies per mil, similar median. And very high, the high loads published, 9 × 10⁻10 RNA copies per mil, and the low 1.3 RNA copies per mil. So very similar to what they found, right?
When you said that, so the mean, at least for the student samples, was 2 × 10⁷, and the median was 2 logs lower than that.
Yeah.
So that means that there's a bunch of individuals who have a lot more virus.
Yeah.
That's shifting the curve in that upward direction.
Yeah, like that Supershedder, right? That one person can really shift the curve. And in fact, I think that's what's going on here when, 2% of the individuals have 90% of the virus. Hey, it's a couple of Supershedders. Very hard to say 3 times fast.
I want to interject here that we don't really know what an infectious dose is. No. I've only seen, not that I've dug deep, but I've only seen one paper back in the SARS-CoV-1 days that comes up with a suggested infectious dose of about 250 active virus particles. I think it was active virus particles, not genomes. And of course, we don't know how many of these, what's virus here and what's just RNA. So we don't really know out of this what an infectious dose is.
Right. All right. So they basically conclude that the distributions of virus RNA levels in asymptomatic and symptomatic patients are quite similar. Similar distributions of saliva viral load regardless of symptomatic status. And they note this has been also seen in studies of anterior nasal or nasopharyngeal swabs. And that's part of the issue here that, you're asymptomatic and you're shedding a lot. And that's why it spread so easily, one of the reasons. All right, so then they ask, how is the virus distributed? between individuals, and they can look at these data and get that information. And that's where they find that just 2% of individuals have 90% of the RNA copies. And that's true in their study, their university asymptomatic study, and also in the hospitalized populations. And 99% of community circulating RNAs are accounted for by 10% of asymptomatic and 14% of symptomatic population. Wow, that's really amazing. So there's a really asymmetric distribution of viruses in both of these populations. And they say, we don't know if that is because these people are special, right, at either extreme, or whether many people pass through a period of high load and we're just not capturing them here because, as Rich said, this is a single time point. I mean, that would be really interesting, a study to do, right, is take this kind of study and find positive people and they say, hey, come back for 10 days.
Yeah.
And it would really give us some information about the idea of super spread. I know that we talked about a paper once upon a time, that talked about super spread and some individuals spreading a lot. I remember a figure with a singer who went to multiple bars or a wedding and showing a map of the spread there. Yep. And we talked about whether or not this was about the individual being special as a super spreader or whether the individual had just the right circumstance. Yeah. They happened to have done an activity that allowed them to super spread. And here, is that one person they catch with the very high number of viral copies unique, as you say, and a super spreader? Or are you just going to be a super spreader if it so happens that on the day you have that your high level of virus, you happen to go to a bar? And so it sort of hits at that same question.
They do say, so there's, we're kind of going around this in a way that's maybe not as direct as they say in the discussion. They're trying to explain why there's these differences among individuals. And maybe it's because you're catching them at various times in their infection. You're catching some of them early, some of them middle, some of them at peak. But then they make a point saying, and they reference several papers showing that peak viral loads do vary dramatically between individuals. So it does lend credence to the idea that there is some subset of people who really are very high viral load. And if they happen to go out and go to many bars and sing, they could become that super spreader.
They note that infectious virus particles, now they use infectious virions, that's good, have rarely been isolated from clinical samples of individuals with a viral load of less than 10 to the 6th RNA copies per mil. This is maybe the sentence that bothered Brianne, right? As written, infectious virions have rarely been isolated from clinical samples of individuals with viral load less than 10 to the 6 virions per mil. No, not.
Yeah, I was very confused.
10 to the 6 RNA copies per mil. And right. So that means for their results, half of the people who test positive may not be infectious to others based on the extrapolation from that. Half. That's interesting, isn't it? How many people were unnecessarily quarantined early on in this outbreak? Oh, well, we didn't know. Yes, so they say a strong implication is that these individuals who are viral supercarriers may also be super spreaders. And they cite the paper that Brianne mentioned that we talked about. I think it was a study in Hong Kong where, among others, 80-90% of infections are caused by 10 to 20% of infected individuals. So that would be consistent with this idea, as Kathy said also. They also say something interesting. A higher rate of spread by viral supercarriers would also be consistent with the surprisingly low transmission rates reported between roommates, schoolmates, and household members, which could be explained if only a small fraction of individuals have high enough viral loads to facilitate active transmission.
That's interesting because that takes into account, in a way, this time factor.
Yeah.
Because if you have somebody that's positive and they're around a roommate for a long period of time, then they get through their whole infection. And if everybody goes through a high viral load phase, you would expect a high probability of transmission to your roommate. But if that's not something everybody does, then you might expect a lower transmission to your roommate. And they're saying it's the latter.
Yep.
Interesting.
That's a really good point.
So they favor, as Kathy said, that they call it the parsimonious explanations, that individuals produce different levels of virus. And, why? Who knows? I mean, everybody's different. So immune responses, other things. So maybe even initial infection site or dose could influence, how many cells are infected and therefore how much virus you make. So all interesting things.
This reminds me of us showing data in our joint lab meeting with infected mice that are inbred and are supposedly therefore genetically identical. And you get different results. And molecular virologist in the group said, why is that? Why aren't those mice all the same? Aren't they inbred?
Yeah.
So, and with people, we're not even inbred. So there's a lot of variation.
Yeah. So they say these results are actionable. Let's do community screening to identify supercarriers. Wow. And then maybe that would have, a bigger impact on curbing infections if you could identify supershedders. I don't know how practical that would be, right?
Well, if you just had, once again, I don't know how practical it is either, but if you just had the quantitative result
and quick, you do turn around quickly,
the CT value, then, somebody, and you've got these data, then somebody who comes in with a huge viral, there may be a bunch of people that you don't even have to quarantine, and a bunch of others you say, whoa. Yes.
Well, actually, you're right. We never got to the point where there was widespread quantification of sample test results, right? And I think if you could do that and then get it turned around in 24 hours or less, that would say, hey, you have a high RNA copy load. Why don't you stay home for 10 days, No, I'm not staying home. Goodbye. What are you going to do, right? I don't know how you enforce it, but that's not my cup of tea, bowl of wax, whatever. I don't do that. Other people can figure out the sociology.
Yeah, you've got to also figure out a way to make sure that you're not really stigmatizing that person.
Well, if it were me, I don't mind being stigmatized because I understand that I'm genetically predisposed to making high RNA copies. That's fine. But yeah, other people might get stigmatized, right? And there's not many virologists in the world, so what are you going to do? Anyway, I thought that was an interesting study, and I have to point out the two last authors, Roy Parker and Sarah Sawyer. This is not what they do. They do other viruses. In fact, Roy Parker does stress granules in virus-infected cells, and Sarah does host virus conflicts, but they have repurposed themselves to doing SARS-CoV-2. This was cool. All right, now we have a paper, which is an article in Nature. It is called Neutralizing Antibody Vaccine for Pandemic and Pre-Emergent Coronaviruses. And this is a topic that has gotten now a bunch of attention. There have been some other papers of similar ilk. This one has a lot of authors from Duke University, University of North Carolina, NIH, FDA, University of Pennsylvania, and BioQual. And they include, so on this paper, let's see, the last author is Barton Haynes, and then the second to last is Ralph Barrick. And let's see who I can spot in here that we've had on TWiV besides Ralph. I can't see anyone else. First author is Kevin Saunders. Second author is Esther Lee, just in case. A lot of authors. So the idea here is, can we make a vaccine that would protect us against not just SARS-CoV-2, but other related coronaviruses and maybe future coronaviruses that might emerge zoonotically. And we know that we can make cross-reactive neutralizing antibodies. So SARS-1 infected people have antibodies that can neutralize multiple related coronaviruses. So you know that these are possible, right? So that's what they explore here. And
just to make sure that people understand this, that doesn't mean that if you get infected with SARS-CoV-2, you're immune to SARS-CoV-1 or one of these other things. What it means is that if you look closely enough, among the many antibodies that you make in response to a SARS-CoV-2 infection, you can find one or a couple that will react with the spike protein of and in fact neutralize, at least in laboratory conditions, will neutralize these other viruses. It doesn't mean that you're cross-protected, but you might be able to take advantage of that to make a vaccine that will give you cross-protection.
Yeah, so that's a really key point here is that, in fact, this is a big part of what the Haynes Lab does, is look for those sort of rare neutralizing antibodies that are made during different viral infections. And they might be rare in that they're made at low levels in most people, or rare in that only a very rare individual makes them, and try to figure out, okay, so this is the antibody that a lot of people don't make. How can we use a vaccine to induce everybody to make this antibody?
Yeah, such broadly neutralizing antibodies have been found in influenza infected people and HIV infected people. And then, the key is how do we induce these? Because they're pretty rare.
Yeah, right.
And that's partly what we're going to look at here. Because, it does.
And Brian,
go ahead.
Sorry.
It's okay.
Yeah.
Brian, help me out here. I have this sort of vague notion that some of these antibodies have really wild structures that have sort of like extra long loops in their binding regions that can kind of snake in and interact with antigens in unusual ways. That's as good as I can do. Am I anywhere close?
So for the HIV broadly neutralizing antibody, that's pretty much correct. So basically, what happens is that after, when the B cell has seen the antigen and starts to make the response, it picks up some additional base pairs to lengthen the region that is binding to antigen. And so it has basically some additions of base pairs to get extra information and make an extra long loop like you're describing. Okay. And so you would have to sort of luck out in terms of having those correct added base pairs to make that extra long loop.
Okay. Crazy.
So here in this paper, they're going to look at whether we can induce such antibodies in primates. All right. Because people have done other studies in mice. Now, they note that a target of cross-neutralizing antibodies is the receptor binding domain of the spike, just the part of the spike protein that binds the receptor. And they say one of these antibodies, remember this number, DH1047. That antibody, which reacts with the RBD, cross-neutralizes SARS-CoV-1, SARS-CoV-2, and some bat coronaviruses. So this is like, hey, this is what we want to be able to do. Now, they also note the RBD is going to be the focus here. And they say we want to make the RBD even more immunogenic than it is. And we can do this by putting many copies of it on a nanoparticle. Think of a small synthetic particle, and we're going to put lots of copies of RBD on it. And they say this, mimics a virus-like particle. Right, it's sort of like a virus-like particle instead of having just proteins floating around, or maybe even florets attached, a few attached by the C-terminus. So you can have a lot of proteins here. And this is the cool part of this paper, which blew me away. So they construct Rbd nanoparticles using a technology that I'd never heard of called a sortase. Sortase is an enzyme from prokaryotes that's normally used to put proteins on the surface of the bacteria basically joins two proteins together. It recognizes a signal at the end, at the C-terminus of one. It cuts it and then brings them both together. And they have taken advantage of that to modify. You can modify your, say, two proteins you want to join together. You got a nanoparticle and then you got RBD. You can modify it so that you add the sortase and boom, the RBD gets attached to the nanoparticle. How cool is that?
So I'm going to make up a story here, and some bug people can write in and correct my story. But the way I imagine this happening, based on how it's described for bacteria, is that a protein that it wants to be permanently stuck on the outside of the bacterium, anchored to the cell wall, is made as a transmembrane protein. Okay. And because it's got a transmembrane domain with this sortase site in a situation that I imagine would be just on the surface of the membrane in, I guess, upstream, if you like, of the transmembrane domain. And then the sortase, in the final step, clips that sucker off and moves the whole peptide to a permanent anchor position on some sort of polyglycine bit on the cell wall.
Yeah.
Incredible.
That's amazing. And this has been repurposed to make nanoparticles. I'm sure it's been repurposed for a lot of things. But essentially, you can make your nanoparticle, which you start out. So the nanoparticle has, I think, oligoglycines, right, on the surface. And then the RBD, you put the signal at its C-terminus, you add the sortase, and it's conjugated to the nanoparticle.
I had a million questions about this.
But isn't it great? Isn't it amazing?
Yeah.
Yeah.
This concept of clustering epitopes on a nanoparticle seems to come up again and again.
Yeah.
Okay. And I had an interaction with a papilloma virologist some time ago, and he actually wrote one of these PLOS pearls about it, suggesting that for a lot of vaccines, having a dense clustering of epitopes on a like a particle, like a virus particle or some other surface, is advantageous for a vaccine.
Yeah.
They ought to do this for HIV, Brianne, because they're very sparse, the glycoproteins on the particle, right? But this would get around that. Absolutely.
And the other thing that I'm kind of wondering about this is the first thing that sort of blew my mind about reading this paper was whenever I've thought about, broadly neutralizing or antibodies that could sort of be pan coronavirus, I've always thought about maybe having to make a synthetic RBD or part of the spike that seems to represent many different coronaviruses. And the fact that they are using a part of just SARS-CoV-2, they didn't have to do any design here, was really interesting. And then I also wondered, could you use this sortase? So it sounds like you have this particle with 24 RBDs on the outside. Could you make a version of it where you had mixed RBDs?
Sure.
And then you could have multiple different antigens and sort of even increase your breadth. I don't know about the sort of biochemistry of it, of how reproducibly you could make the same variant versions of it, but I thought that could be really cool.
My interpretation of this is that the RBD is like a platform, and then there's decorations that make it hit one or another receptor, but the framework has got the cross-reactive residues, because otherwise, this RBD hits ACE2, right?
Right.
But other coronas are going to hit other receptors. So how do you hit those? So maybe there's a framework and then there's a variable part.
And this must be there must be some conservation in either the sequence or the structure of the RBD across beta coronaviruses that allows this to happen, to get this cross-reactivity.
Right.
Yep. So they put 24 RBDs on these particles. They confirm that they've made the particles, and they test them. As you would guess, these particles bind ACE2. They also bind antibodies that bind the RBD of SARS-CoV-2, which is what you would predict. They also, and there are a whole bunch of these antibodies with DH numbers. And DH 1047, remember, folks, that's the cross-neutralizing antibody against the RBD. That also binds these RBD nanoparticles. So it looks like it's structurally okay. So then, you inject it. You immunized macaques, cynomolgus macaques, which, by the way, Albert Sabin used to develop and test his OPV strains many years ago. They immunized them intramuscularly with an adjuvant. Apparently, the macaques didn't complain. And they make antibodies that bind the RBD, that also bind the spike, the proline prefusion stabilized spike protein. So the antibodies that these macaques make both bind the nanoparticle and the spike itself. And what else? They
compete with binding of spike to ACE receptor,
right,
or binding of spike to some of these antibodies.
Right, and they did neutralization using pseudoviruses, SARS-CoV-2 pseudoviruses that induced, this is pretty cool, in neutralization titers ranging from 21,000 to 162,000. Wow, that's a really high neutralization titer. I mean, if something's 50,000, I think it's pretty good, but 162,000. And they compare these titers to those from macaques immunized with basically mRNA lipid nanoparticles that encode the prefusion spike. It's basically the vaccine that many of us, Pfizer, BioNTech, Moderna, and those make nice neutralizing antibodies. But if you look at the geometric means, so the RBD nanoparticle, 47,000 versus 6,500 for the geometric mean of the neutralization titer of the lipid, of the RBD nanoparticle versus the mRNA vaccines. So get higher neutralization titers. So they say
higher.
Sorry,
by geometric mean, about 8-fold difference in the geometric means.
And even higher neutralization titers than convalescent serum.
Yeah, I think that was really important.
Yeah. All right, so then they look at virus. They look at B117. They're looking at B1351. They look at P1. And these of course have spike amino acid changes in the RBD that it can impact neutralization with monoclonal antibodies. And
think variants. These numbers are variants of one sort or another.
So there are 3 amino acid positions, 417, 484, and 501. These are, they say, distal to the binding site for DH 1047, which is the broadly neutralizing antibody. But 484 is within the binding site of 1041, another antibody. And so they say, this is very funny, 1041 binding to SARS-CoV-2 RBD was knocked out by the E484K mutation. It's a funny word to use, knocked out. But, scientists like to use things like abrogated or impacted. I mean, it's fine with me. I'm just surprised. But for 1047 binding was not affected by any of these 3 amino acid changes. 1047 is the broadly neutralizer, right? And so these changes are apart from its binding site, so they're not affected. So they asked whether the macaque sera how the macaque sera did against these variants. So B117, no problem neutralizing it, but also mRNA vaccine had no problem neutralizing B117 or its ancestral virus, which they call D614G. And then their RBD nanoparticle sera neutralized all these guys, all of them, with they neutralized all of them. Actually, both the mRNA and the receptor binding domain nanoparticles immunized macaque sera neutralized all of them, although the RBD particles were a little more potent, higher titer.
Yeah, I liked to be able to see this data because these variants that they're listing are the ones you've heard about in the news with different geographic names, and it was comforting to see that. Look, the mRNA vaccines do neutralize well. We don't know what is the protective neutralizing titer to know if we've gone above or below it, but we can certainly see the neutralization is still there. But the fact that there really is no loss of neutralization with this RBD is pretty impressive.
Yeah, it does better than the mRNA induced sera. But and of course, remember the significance of this is not entirely clear. What's really clear is protection against disease, right, in a person. Mm-hmm. And as we have seen, that some of these, you can have a tenfold reduction in titer, like the J&J vaccine did against the South African variant, and still protect against serious disease. So this is not the whole story. I say that because people freak out all the time. I hear people freaking out that the antibody titers are low, we're going to all die, but it's not the whole story. It's not the whole story. So both vaccines unaffected by changes in the B117, but the other two changes had some effect, but the nanoparticle induced sera did better than the mRNA, although the mRNA is still neutralized. Is that a fair summary of that?
That's a fair summary.
Yes.
All right. So then they said, let's look at some other SARS-related coronaviruses, SARS-CoV-1, and then a couple of SARS-related bat coronaviruses. The names are not going to mean anything to you. WIV1, that must be Wuhan Institute of Virology 1, and then SHC14, which I remember Ralph Barrick's lab working on. And then they said, can these be neutralized with sera vaccinated with either the RBD nanoparticles, the mRNA? Now, I think they make these for each of these RBDs, right? I spent a long time trying to, and I think that's what they do. They take the RBD of each of these viruses and make RBDs and mRNAs, and then they look at cross-neutralization among all of them.
I think that's correct.
And so they compare again RBD nanoparticles with mRNA vaccines, and again, just like before, the neutralization titers are higher with the RBD compared to the mRNA. They get cross-reactive antibodies. They differ depending on the virus used for immunization, but they're all induce antibodies using this RBD approach that neutralize the other viruses. So we're talking about SARS-1 and the two bat viruses. And also SARS-2, the SARS-CoV-2 RBD will also induce antibodies that neutralize those viruses as well. So they say the SARS-CoV-2 spike induced cross-neutralizing antibodies against multiple group 2B SARS-related beta coronas with the highest titers induced by the RBD nanoparticles, which is good. It's really good. How it translates to challenge, we'll have to see. They're going to do that.
Yes. So they do show that, this isn't unlimited cross-reactivity. So we don't have the cross-reactivity against the MERS RBD. And so, this is not necessarily going to be a completely pan coronavirus type of thing, but it certainly gets us more breadth than we've necessarily had before, and is something perhaps we should have been thinking about before.
So this immune plasma did not bind to spike from the 4 endemic coronas or MERS coronavirus.
So I'm looking at a slide that we've used before. That's phylogeny of coronaviruses that splits out, shows the relationship, phylogenetic relationship between what are called alpha coronaviruses and 3 different lineages of beta coronaviruses, A, B, and C. And whereas SARS-CoV-2 is in lineage B, as is SARS-CoV-1, MERS is in lineage C, and the human coronaviruses are either alpha or in lineage A. So they're in different arms. The ones that are not neutralized are in different arms of the phylogenetic tree. How that relates specifically to the receptor binding domain, I don't know. Be nice to see an alignment and an analysis of this.
And the way I was thinking about it is that MERS, the receptor for MERS is DPP4, completely different molecule from ACE2. So that didn't surprise me.
Yeah. Now the common cold coronas, do any of them bind ACE2?
I believe one of them at least
one of them
does.
does. Yeah. So
I can get that back up for you.
So that it's surprising that you don't get any cross neutralization. So maybe the cross neut is not just against the receptor binding domain, or maybe it's different enough.
Right. And in fact, I think they state that as one of their possibilities, or
I don't have the answer to that question right at my fingertips.
So there's I don't know if the figure you looked at has the group the subgroups, the group 1, 2A, 2B, and 2C on it, because that's another way that they group these coronaviruses. And there's actually a figure in this paper where they do that. But the 2B related, the group 2B coronas have the highest cross-reactivity induced by the SARS-CoV-2 spike. All right, so now another experiment. I hope I can get this right. It's a little bit complicated. So DH1047 epitope, that's the epitope that induces antibodies that can cross-neutralize a number of these beta coronaviruses. So that's in the RBD, right? It's right near the N-terminus of the ACE2 binding site. And they say it's different from other ACE2 neutralizing antibodies like DH1041. It's a little in a different place. And DH1047 is highly conserved among these group 2B coronaviruses. So they're asking, can we induce DH1047-like antibodies, essentially, by doing competition experiments? Right? They do blocking assays. Plasma from all the macaques immunized with RBD construct can block ACE2 binding, as we said before. They can block DH1047 antibody binding to the spike. All right, so that's their assay that they're using. Those antibodies, the blocking antibodies in the RBD sera, the RBD nanoparticle sera, are cross-reactive because they can also block DH1047 binding to a bat coronavirus. 5 of the 5 vaccinated macaques have very strong DH1047 serum blocking activity. In other words, they have made DH1047-like antibodies by immunizing with these RBD nanoparticles. Also, 3 of 4 immunized humans and mRNA vaccine immunized humans, and 9 of 22 convalescent human sera had DH1047 blocking activity. So that antibody is also present there, they say. So this means that the 1047-like antibody response is subdominant in infected or immunized humans, because it's only 3 of 4 or 9 of 22, and also in mRNA immunized macaques. But it's dominant response to the RBD nanoparticle vaccination, which is good, right? You want it to be dominant. You want it to be against this cross-reactive epitope, which was an issue we talked about earlier with these rare, broadly neutralized. How do you get them induced? Well, apparently looks like this is working, right? By making these nanoparticles. Yeah.
And there also seems to be a difference in the kind of amount of those antibodies that are made between the groups. And so not only is it just yes or no, are you making and one of these antibodies that blocks DH 1047, but there's also a difference in the amount that's being made.
Yeah.
And so the macaques that are getting this nanoparticle are also making considerably more of this because it's the dominant epitope in this case, instead of being subdominant in the other cases.
So this, as I was reading this is a good place, I think, to bring us up. As I was reading this, I was remembering conversations that we had months ago about the different vaccines that were being made. And some people were making vaccines that were to the full-length spike. And there were a couple of vaccines that were being made just to the receptor binding domain. As a matter of fact, Pfizer made initially 2 mRNA vaccines.
That's right. Okay,
yeah. One of which was to full-length spike and one of which was to the receptor binding domain. And the full-length spike seemed to work better for them, so they dropped the other one. Okay.
Yeah.
And we said, oh, you're crazy doing full-length spike. I mean, doing receptor binding domain because you can make more antibodies to the full-length protein. And this is sort of the flip side of that. It seems to me like, by presenting in particular in a nanoparticle,
that's right,
the receptor binding domain, you're telling the immune system, focus, dude. Focus on this.
That's a good point.
Okay.
Yeah.
Does that, Brianne, does this make sense that the full-length protein in a way would be a distraction? I mean, you're talking about dominant and subdominant epitopes. Is that what I'm talking about?
That certainly is the case, is, that you are basically focusing the immune response on the epitope against which you want to have a response, and you are potentially excluding other epitopes that may dominate over the one you want, so that you get the one you want to be dominant.
Okay.
Of course, part of me, and I mean, so this is the lab I actually worked in as an undergrad. I know quite a few of the people here, and so I love everything they do, although there's a part of my head that's saying, but what about T cells? Right now.
Yeah.
And so, that would be sort of the downside, is that you're limiting this to making a really good antibody response, but you may not make some of the other types of immune responses. And so that's why it's really important that they also look at things like challenge.
Yeah. Of course, if you have a sterilizing antibody response,
then you don't care.
Then you don't care about T cells, but
exactly.
I don't think you should, as Sete said, I always want to have both. But Brianne, so I'm looking at this in 2 ways. You have a nanoparticle with a lot of copies of the epitope, right? So when that gets taken up into APCs, that's all they see, right? That's one thing. But what about B cells? Aren't B cells going to see this multimeric thing? Isn't that going to play a role?
Yeah, they are. And so your B cell is going to be more likely to have 2 receptors on its surface, be cross-linked to give that B cell a better signal and better activate that B cell.
Okay. So it could be at 2 levels. All right, good. All right, so then we do a challenge experiment. They have 2 groups of macaques, right? Nanoparticle or mRNA immunized. And they infect them intratracheally and intranasally with 100,000 plaque-forming units. And these then they say, what's going on? They can find infectious virus in their unimmunized controls, which shows you that the infection works. And then they do PCR. On day 2 after, they challenge them, of course. I already said that they challenged them. I'm sorry. On day 2 after challenge, the unimmunized macaques have an average of 1.3 times 10 to the 5 and 1.2 times 10 to the 4th copies per mil of the E subgenomic mRNA in nasal swab and bronchoalveolar lavage fluids. Okay, so that's kind of the baseline. And then vaccinated monkeys, they can't detect any E subgenomic mRNA in the upper and lower tract. Nothing. That's amazing, because PCR is very sensitive. And then they look 2 days later, and again, no detectable RNA in any of these samples.
I gather that in this particular animal model, the animals don't get sick.
They don't get very sick, no.
And that's why they're doing these sort of molecular diagnostics for infection.
They also looked in lung tissue for viral antigen nucleocapsid protein. They don't find it. but they do find it in the control macaques. H&E staining of lung tissue showed a reduction in inflammation in immunized macaques compared to control. So there is some inflammation going on there, which you would expect, but there's no virus production. They also looked at mucosal immunity. They take bronchoalveolar lavage, they concentrate it, and they get the IgG out of it, and they can show that this will bind spike and block ACE2. It will also block these antibodies binding to spike. So the immunization not only seems to protect from infection, they call this sterilizing immunity, but it also makes mucosal antibodies, which is probably important for blocking infection entirely, right? So yes, go ahead. Sorry.
Well, it's a digression.
That's okay.
If you want to. So in the vaccine town halls discussion group, task force, it came up that using the term sterilizing immunity, maybe we've even talked about this on TWiV, has led perhaps some proportion of the population to hear people say that and then think about the vaccines leading to fertility issues.
Oh my gosh.
So
yeah.
But it's true, Vincent. If you think about it, the terms that people think about sterilization.
It's fine. No, I think that's a great point. Yeah.
If there's another word that could be used, or phrase, I can't remember if we came up with something in that task force meeting, but it was just like an aha moment. Oh,
yeah.
If you use,
I think in the scientific literature, sterilizing immunity is so burned in.
Yeah, it is burned in.
That it's going to be hard to get around that one. But the qualifying, it does the awareness of the misconception is important, and so the term does need to be qualified.
So maybe we need to start talking about infection blocking immunity or disease blocking immunity.
Sure.
Mm-hmm.
Fortunately, the SARS-CoV-2 vaccines are not sterilizing. It's a good thing on all fronts, right? But that's a good point, and that's maybe how I would not be surprised if that's how all this misinformation happens.
Some of it is from that, and then some of it is from the 3 to 4 amino acids.
Synthesis.
But
wow.
But it reminds me of, the first time I heard about physicians talking about, something or other insult. And it's like they're using an ordinary word, an insult, in a way that's different context. So, yeah.
So here are some words we could use instead of sterilize. Oh my God, they're all terrible.
What do you got?
Well, I'm looking at a
thesaurus.
I'm looking at a thesaurus.
Yeah.
They're all terrible. Disinfect, purify, fumigate, decontaminate. No. Then they're all to do with, oh my God. Spay is another word for sterilize.
Yeah, actually, when it comes right down to it, in a way, sterilizing is not
a good word.
Not really appropriately descriptive for what happens, right? Something more like what you said, Brian, infection blocking or something.
Infection blocking. We will use that on TWiV from now on. Infection blocking and disease blocking, because they makes perfect sense, right?
And that also kind of highlights that whole protection from infection and protection from disease issue.
Yeah. So that's the story here. I would line up tomorrow to try this
it's really interesting
vaccine. I think, and they say there's no evidence of increased immunopathology in these animals. No enhancement evidence at all. And they say it represents a platform for producing pan-corona vaccines that could prevent, rapidly temper, or extinguish the next spillover of a coronavirus into humans.
What distinguishes this from the Novavax vaccine platform, which is those florets? Is it more, or that it's it's the smaller domain, and it's
the Novavax vaccine is full-length spike.
Yeah,
that's actually not attached to a separate particle. It somehow aggregates by its C terminus all by itself.
How many of them aggregate? Do you know?
I don't know, but conceptually, although it's a different antigen because it's full length spike, conceptually, the idea of clustering the individual epitopes, that concept, I think, is retained, even though it's done differently.
Yeah, and so the only issue is that perhaps in that case, some other epitope dominates over this epitope that they're looking at here.
Probably it's a combination of that and the number of copies. Probably there are more copies here on the nanoparticle, yeah.
I put a link in here to this Plas Pearls paper that I was talking about that talks about epitope density. I have one other question for Brianne. Do you know anything about this adjuvant? 3M052?
So 3M052, basically, all I know about it is that it is a TLR agonist. So it's hitting TLR7, TLR8. So it's sort of an RNA like molecule. And so it's going to give you a pretty good kind of innate immune response that the protein by itself wouldn't give you.
Okay.
It's a nice story, isn't it? I like it very
Great.
much.
It is.
I really like it. And, this is, this is where we need to be going. This is foresight. This is, you need to have this and several other like type vaccines, at least conceptually, on the shelf in anticipation of future spillovers. Okay, so we're not caught flat-footed.
I hate to say it, but this was all doable a couple of years ago.
Sure.
Should have had these already. Oh,
well, it was, although, I will note that they are using part of the SARS-CoV-2 RBD here. And so they would have had to design some RBD to use or find another one that would have worked.
Yeah, they could have used the SARS-CoV-1 or any of these other. I mean, they all do cross-reactivity to a certain extent, right? So I would have tried it anyway with SARS-CoV-1, but the problem is funding, right? It's hard to get funded for that. SARS-1 is gone. Who cares? But maybe we'll have it for the next one. That would be great.
So I have a basic thing that I think I've missed here, and that is how did they pick a certain part of the RBD to be in these nanoparticles by some planned rational design, and does that relate to the 1047, or did they just use the whole RBD?
I believe they used the whole RBD, yeah.
Okay.
Yeah, my understanding is they used the whole RBD, but then they found that the broad neutralization was in the antibodies that could bind specifically at that site, the 1047 site.
Yeah.
Okay.
They certainly were interested in using, I don't know how this went into the design, but they were interested in using a portion, at least, of the receptor binding domain that would be reactive with several of a panel of cross-neutralizing monoclonal antibodies.
Yeah.
Okay.
Yeah.
So you're not just focused on one, you're focused on several. In the end, they focus more specifically on one, but you start off with a panel of monoclonals with different cross-neutralizing specificities.
Right. Protein
Great stuff.
production. Yep. RBD. They use the whole RBD.
Yeah, because the problem would be that if you start to decrease that RBD more and more, eventually you're going to lose the folding that you might need, so that you might not get the actual epitope to fold the way it needs to. And so the rest of it is just the framework around which that folds. And at some point you can't get any smaller and still have the folding work.
So here's a question. So if let's say you made this vaccine, you immunized 5 billion people, would you get variants that could change in these residues, even though we haven't seen them so far? Would that put pressure on these conserved
Well, I was
residues?
wondering about that because if there are portions of an ACE2 binding domain that are illicit cross-reactive antibodies,
Yeah.
are those elements that are essential to the ACE2 binding so that you can't?
Good question.
There's no workaround for the virus. I don't know.
We could do that experiment in the lab. You could do passage with the antibodies and see. Yeah, that's a good question. Maybe it would change, maybe not. So the reason I ask is because a certain virologist who will not be named has said that there is no residue in spike that can't change, which I just don't buy.
Well,
There's always a residue.
Yeah, I mean, you look at the supplemental figure where they have the multiple sequence alignment. There's long stretches where in all these different coronaviruses, things are identical.
Yeah.
You know, yeah.
No, I think there's always framework residues in these spikes that cannot change. So anyway, that would be interesting to see if you could take this, well, maybe someone's done it, this 47 antibody and passage virus and see if you can get resistance. Probably it's been done. I just haven't seen it. Cool stuff. Anyway,
maybe we'll get mail.
Yeah, we can get mail. That's how we don't know everything, folks. And I get a lot of email from people who say, why don't you look things up before you say them? Well, that's not the spirit of a conversation, is it? When you talk to someone, before you say anything, do you go to the library and look it up before you say it? No.
You know, I don't have all the ideas in my head until I talk to you guys.
Right, exactly.
Okay.
That's the point.
I wouldn't know what to look up.
And I'm trying to figure out sort of how that mutation would work if these antibodies are sterilizing or infection blocking, because you'd have to have infection to allow for mutation to happen. And so I'm sitting here trying to work that out in my head. And
part of science is to speculate and muse on things, right? And I think saying don't say anything unless you look it up is just contrary. I get that all the time. Don't say this until you look it up.
And we don't have infinite amounts of time. You know, we read these papers and we discuss them. And
however, if you'd like to pay us handsomely, I will give you infinite amounts of time. But nobody's going to do that. So,
so I have a teaser for my pick of the week, which is a quote from Richard Feynman. He says, I can live with doubt and uncertainty and not knowing. I think it's much more interesting to live not knowing than to have answers which might be wrong. I don't feel frightened by not knowing things, by being lost in the mysterious universe without having any purpose, which is the way it really is, as far as I can tell, possibly.
Yep.
So, you know, not knowing is okay. As a matter of fact, that's what we're all about.
That's how you get to learn things.
Yeah. When you know something, you're done. You go on to the next not know.
In fact, look, if something comes up and we haven't read the paper on it, we're going to talk about it. And you know what? If we're wrong, just tell us. I don't mind listeners writing in and correcting us. That's fine.
Yeah, that's great.
All right, let's do some email here. Kathy, can you take that first one?
Sure. Carl writes, hi, I'm the chief operating officer of a big European direct-to-consumer media and communications company. I lead our work to keep our staff and customers safe through COVID. Oh, maybe that was I led our work. Guess so, spelled lead. TWiV has been brilliant at giving me better context to make risk decisions for our day-to-day operations, including our approach to working in customer homes and workplace testing. If you had told me 12 months ago that I'd be listening to a podcast on viruses, I'd never have believed you. But that's not what I'm emailing about. In episode 747, you mentioned the $25 antigen test now available at pharmacies in the U.S. I thought you might be interested to know that the National Health Service here in the UK has from March offered self-swab antigen tests for free to anyone who wants them. They're distributed through workplaces, pharmacies, online, and also through schools where a twice-weekly test is mandatory for everyone over 12. The tests used are produced by Innova and SureScreen. The NHS recommends that everyone test themselves twice per week. Results should be reported through the NHS COVID-19 app, and positive results are followed up by a free PCR test, which determines whether continued quarantine is necessary. Carl.
That's great.
So yeah, my cousin lives in London and has a border, and so my cousin is my age, and her boarder is quite a bit younger, and went to get some of these tests and begged for more than her share because she lives with these really old, decrepit, very feeble, susceptible people. So she got like 30 of them at once or something. But we had a good laugh about that. So,
National Health Service.
Yeah.
My goodness.
Yeah. Well, that's one of the benefits, right?
mm-hmm.
Never going to happen here, is it?
Well, not probably in my lifetime.
Yeah.
I keep
too.
I keep hoping. But at any rate, good on you, Brits.
It's
Yeah,
good.
this makes it sound pretty good.
That's good. Brianne, can you take the next one?
Sure. Alex writes, I just had to pause TWiV 754 after hearing your listener's email about a weeks-long migraine following vaccination. These symptoms are consistent with the rare clotting reported in females after being given other vaccines. Clots blocking the sinuses cause pressure buildup in the blood supply that drains into the blocked sinus. Maybe the pre-fusion stabilized spike protein in the vaccine she had reduces the chance that this can happen. So we haven't seen deaths following the mRNA vaccines. But you just read a paper about the effects of the E protein in a related mouse virus. One could easily imagine a possibility that the spike in humans could have some other effect, even if rare. Unless somebody who knows something can explain that I am wrong, very possible, I have no experience in the field, please advise listeners to take post-vaccine migraines more seriously, especially in the high-risk group, females 18 to 49. Ibuprofen is probably a good call to reduce inflammation. Also, remind them to use the vaccine adverse effects reporting system so the right people can pick up if there is any signal. Facebook groups are not a substitute. Now back to the video, Alex.
Can someone speculate about clots? So Alex thinks maybe the vaccine induced these clots, right? Is that what he's saying?
Yeah.
That's what I'm getting.
He's referring to, I think, the adenovirus vaccines that were giving the rare clotting things, and that one of the clinical signs of that was severe headaches.
I think this lady who wrote in had a migraine after an mRNA vaccine. That's why I'm asking.
Correct.
Because I don't know how.
Correct.
I mean,
I think Alex is speculating that maybe that this could be the case for other vaccines.
So, I guess my question is, if you get an intramuscular injection of any kind of vaccine, does some of it end up in your brain? Is it likely?
It's hard to imagine that happening.
That's what I think. I mean, most of it is staying in your arm muscle, right? But
yeah, I mean, from obviously I'm sitting here thinking about some of the experiments we used to do in graduate school where we would try to follow the antigen. And the assays that we were doing wouldn't, show if one single molecule of antigen made it elsewhere, but the antigen was really localized with these types of things.
Brian, that's a great name for a kid's game. Follow the antigen.
That is a great name for a kid's game.
But this, he's not talking about the antigen going to the brain. He's talking about clots in the sinuses leading to headaches.
Oh, so the clots could form somewhere else.
Yeah, so I think he's talking about excess clotting.
Got it.
Yes.
And those clots getting stuck somewhere.
All right.
Well, I think, the important message here is if you've got something other than a background apparent adverse effect, use the vaccine adverse effects reporting system. Talk to your doctor. Okay. Take that stuff seriously. I'm totally on board with that.
Yeah, it's fine. I'm just trying to figure out what's going on.
You haven't talked to Daniel about this long recovery from vaccine.
I did. I read the letter actually on the clinical update last week because I said, maybe you could shed some light because we couldn't. And he didn't. He basically said, hang in there, He said, you'll get better. He didn't give any insight. No, because he doesn't know. Yeah.
Right.
I don't think anyone knows. Let me take this next one from John. Not sure if I sent you this one before, but it would have been a great addendum to the paper discussed in 753RE vaccine-related polio outbreaks. This is a short reminiscence from the son of the head of a prominent Pittsburgh nursery landscaping business, still going strong. Jonas Salk was one of their clients, and they had the bad luck to be working next door on the day he proved that his vaccine worked. Especially everyone in polio research should know this story. Yeah, this is a great article about that. Exactly. I remember reading this. Bullet-nosed Studebakers were the 50 and 51 models. So this is, where is this? The Post-Gazette, which is a Pittsburgh Post-Gazette. The headline, local landscapers had close brush with Jonas Salk breakthrough. Typical Holy Thursday in early April 1955. Yeah, this guy's sitting at his kitchen, and I think he goes outside, and somebody drives up really fast. The door flings open and screams, and that's Jonas Salk, who just found out that it worked. And the funny part here is at the end. Where is it? Pittsburghers know the rest of the story. Where was it? There was an Italian part here.
The driver made a hard left, a quick right, and shot up the driveway right next to us, totally out of control. He just missed hitting one of our workers, Dominique Venezzi of Abruzzi, by the slimmest dusting of his thick corduroy pants. That sounds like the Italian part. And then he answers back in Italian a little below that.
He says,
and then I like the next part.
Donna looked at my dad and said, Max, che tipo di lavoro fa? What kind of work does he do? Oh, that's translated there, but I just translated it on my own.
And then he says, he's a doctor at the University of Pittsburgh, and the answer back is apparently in Italian, although it's translated here. Whatever he does, I guess he just got a big raise and he can buy me a new pair of pants.
It's funny. He just missed hitting one of our workers by the slimmest dusting of his thick pants. Okay. Anyway, it's a funny story.
Good story.
Rich, can you take the next one?
Alec writes, first of all, thanks to all of you again for all that you do and have done throughout the pandemic. I think I may have mentioned this before, that I actually started listening to TWiV while I worked in the Middle East during MERS. And then, like now, your podcast has been an island of objectivity and common sense in a sea of disinformation. There's a episode title. I pray and hope every day that more people will listen to you instead of the mass and social media. I'm writing to ask for your thoughts on the following. I took the AstraZeneca vaccine here in Canada last month, as it was the first one available, and I am considered an essential worker. To borrow Daniel's phrase, I took that opportunity to vaccinate. Needless to say, I'm still alive, and apart from a sore arm for about 24 hours, I had no side effects. Unfortunately, as you may be aware, the AstraZeneca vaccine has had some bad publicity bordering on hysteria, in my opinion, about blood clots post-vaccination. The provincial government here in Ontario, Canada, has decided to stop giving it as a first shot and has announced that those of us who had it as a first shot now have the option of taking it as a second shot or getting one of the other vaccines, which would be Pfizer or Moderna. The study cited for doing this, apparently unpublished, stated that there is a possibility of 0.9 people out of 100,000 people getting a blood clot, which the mass media then decided to round up to one person in 55,000. Interestingly, a study in the UK and one of the largest users of the vaccine found that it was less than half this number from a much larger population. Gives a link. Considering that 5.4 people out of 100,000 in this province run the risk of getting into a fatal motor vehicle accident, I'd say that AstraZeneca is still safer. But hey, that's only me. And it pretty much sums up that saying quote, there are lies, damned lies, and statistics, end quote. My questions are, could there be any adverse side effects from switching to one of the other vaccines for the second dose? I'm going to do these one at a time. Don't know, but I wouldn't necessarily anticipate it. 2, would there be any risk of reduced immunity against SARS-CoV-2 resulting from use of one of the other vaccines as a second dose? I wouldn't think so. Would the J&J vaccine be an option as a second dose? It is apparently available, but I'm not sure on how it's being rolled out here. This I don't understand, because the J&J vaccine, is the prescription is that that's a single dose vaccine. Not that a boost wouldn't necessarily help you. And I think, it could be an option for a second dose, but that's not the way it's been tested and deployed.
Well, and at the time, I'm not sure when he wrote this, but it may be that the J&J at that time didn't have the connection to the blood clots that the AstraZeneca did.
Yeah, maybe.
And so he was looking at that as a similar adenovirus-based vaccine. Yeah, maybe.
To be honest, I will take AstraZeneca as a second dose if offered, given that I'm pretty sure that I am. Oh, I see. He took AstraZeneca as the first dose, and that is a 2-dose thing.
Yes.
I'm sorry. I misunderstood that. Would the J&J vaccine be an option as a second dose? Yeah, sure. I don't see why not. Anything would be an option as a second dose. It's not the way it's prescribed, but to me, they ought to be mixable. To be honest, I will take AstraZeneca as a second dose if offered, given that I'm pretty sure I am not in the 0.9 out of 100,000 cited, but I do know people who would prefer to switch, given all the publicity around it. Finally, I was sad to hear about the passing of Thomas Brock. Although I never pursued a career in it, my undergrad degree was in microbiology. And I, like pretty much everyone who went down that road, started with his Biology of Microorganisms and had to lug that book around campus. Mm-hmm. Thanks again for all you do, and the best to all of you. Okay, sorry I confused that.
It's okay.
But I think, am I correct that the cases of what in my mind is being called specifically TTS now, thrombosis with thrombocytopenia syndrome, to be specific about what this is, usually is associated with the first dose?
Yeah, that's correct. Yeah.
Is that correct?
I believe so, yeah.
That doesn't, to me, exclude the possibility. Of course, I don't know. It shows up within a couple of weeks, so I don't know that you get to the second dose.
Yeah, that's the problem.
But nevertheless, it's not like a lot of cases have shown up after the second dose.
Right.
So that would reinforce the notion that you could. This is strictly theoretical. That there's no foreseeable downside necessarily to following up with the other adenovirus vector. However, if you survive the first dose, you'll probably survive the second dose with AstraZeneca. But he's saying they may not give it to me. So J&J, would be a reasonable substitute, as would either of the mRNA vaccines, it seems to me.
Okay, Kathy, can you take the next one?
Allison writes, hello, amazing TWiV team. This is Allie from Virginia Beach again, the hockey mom and volunteer epidemiologist. This is my follow-up report. Baseline plus post-dose 1 spike IgG report. 2 months ago, I wrote to you about my post-COVID antibodies. Well, I couldn't resist chasing this down further. I ordered, via the internet, a second antibody test from Quest Diagnostics, a semi-quant IgG test, code 34499. Both results are attached. My first result, 4.5 months after natural disease, was 11.21 on an index that was very vague. Greater than 1 is considered positive, but I was not at all sure what 11.21 meant. I thought that it was at least good. I did some digging, which I shared with you in my last email. Now I have results back on day 21 Pfizer post-dose 1. My result was greater than 20. I was annoyed again because not only was this vague, there is no reference range. My conclusion: people with natural disease, PCR or antigen positive, probably don't need 2 shots. The body saw all 25 proteins of SARS-CoV-2 in the first battle against the virus. The vaccine shows 1 protein spike and boosts immunity quite well to the immunodominant antigen. On the day I got dose 1, I ordered my TWiV sweatshirt so that it would arrive as a reward for going through with 2 doses as recommended. I proudly wear it to the hockey rink now. When I see new faces above the mask, I thank them for being gracious with protocols and introduce myself in case they have grievances or feedback. I figured it's best to keep a wide open door to conversation about all of this as we move through pandemic to endemic SARS-CoV-2. Precautions will modify as we learn more. Last night, a man replied to this introduction by asking, "Oh, are you a PhD or MD?" I paused and replied, "Neither," with a bit of a chuckle. I'm just a boots on the ground epi. I keep trying to learn everything possible through incredible teachers like all of you. I am so profoundly grateful. It's a real joy when I anticipate a question one of you raises. I literally talk to myself while running and pump my fist in the air when I nail a question with a remotely similar response. It's like taking the APs again. Thanks a million, Allie in Virginia Beach. And then PS has nitty-gritty details about one of the tests. The Quest Diagnostics test is the one that an individual can order and pay for directly from the Quest website. It was $119 when it first came out. Now it's $78. If you order the antibody test, this is the one that pops in your cart. You have to pay for it, then schedule the appointment.
So wait a minute. If I read this correctly, we don't have the full data set here. We need a test after dose 2, right?
Yeah. She just reported
she's
dose 1.
Concluding, yeah. She's concluding that she's okay after dose 1, but I want to see the rest of the data.
Right. But if they're only going to give her a result that's greater than 20 and she gets another result that's greater than 20,
then she doesn't know anything.
Right.
I mean,
maybe she'll get a result that's greater than 30.
I think it only tells you greater than 20.
So, Allie, I mean, I think you're a great student and I love it, but I would not conclude from your experience that you don't need 2 shots because we don't know what these numbers mean. And really, we don't know what they mean in terms of protection against disease. So,
right. And they're free. So go get the second shot.
And I think Allie is going to get her second shot. So that's fine. Yeah. But I love it. I love when people get into it like that. It's so cool. Yeah. Brianne?
Mary writes, "Dear TWiV team, it's been a long time since I have written. Still enjoying your scientific chats. I'm on an NIH mailing list that recently shared a summary of 2 studies of COVID-19 deaths with potentially fruitful results. It's at," and she shares the link. I'm only an accountant, but I do tend to notice numbers and expect them to make sense. My take from the article is that the studies use samples from at most 30 victims of the disease. IZAR's, Columbia, 19 men and women, and Regev's, MIT, 420 samples from 11 organ systems, possibly 2 or more organs from the same individual, so less than 11 individuals. Yet in the concluding remarks, the article goes, "The hope is that these vast data sets," emphasis mine, "together with future analyses and studies, will improve our understanding of long-term complications in patients who've survived." Wait a minute. Vast? How did we go from so few victims to vastness? The fact that the study data will be made available to researchers worldwide and the potential for more advances in dealing with this disease are both good news. I hope it is not being exaggerated, and it is just my failure to understand. Your fan, Mary, and Mary's from Washington.
I think they're vast because they're single cell analyses, right? So there are a lot of cells in each patient.
Yeah,
but not.
This is the director's blog from NIH that links to the studies, so you can probably get a somewhat more, precise quantification from the studies themselves.
Yeah, they do single cell. They do RNA-seq. So there's a lot of data generated. That's why they call it vast.
Right.
And there's a vast number of authors on the papers too.
Yeah, there's a lot of data, even though it is not from a huge number of individuals.
Yeah.
And even if it's 420 samples from 11 organ systems, that might be on the order of even 40 individuals, but 19 and 40 does not sound like vast.
Yeah.
I agree.
Mark writes, Dear TWiV team, it's the same temperature in Bangkok as the last time I wrote. Probably have seen this already, but just in case, Mark links to a preprint archival influenza virus genomes from Europe reveal genomic and phenotypic variability during the 1918 pandemic.
Ooh, let's do that paper.
Yeah.
I saw this yesterday, so he's not too behind.
Let's get a. This is a preprint. Let's do it when it's published. Is that okay?
Yeah.
I'd like to do that.
Yeah, because this is a serious question.
Yeah, because we don't have much sequence from that, so this is good. Very
No,
good.
and there's all this stuff about different waves of flu,
Yeah.
and nobody knows what was going on, right?
All right. Continuing, I have a question for Brianne. Why do immunologists seem to prefer reporting antibody concentration levels in AU per mil rather than micrograms per mil? Perhaps I'm old-fashioned or just old, but I was taught that proteins actually have mass. My main concern is not really the use of AU per mil, but that many papers don't link to these microgram per mil units. Please explain if you have a chance. From my readings, it seems that post-COVID IgG serum concentration levels range from about 1 to 150 micrograms per mil after 2 weeks. Is this correct based on what you know? Thanks for your continuing work, Mark in Bangkok. What is AU brand, do you know?
So I looked at this and I assumed he meant sort of U per mil or units per mil. I sometimes see IU or international units.
Yeah.
So I kind of assumed that he meant units per mil. And to be honest with you, I find units per mil incredibly frustrating. I come across it more often when I'm thinking about cytokine measurements. But what I have learned is that this is because different manufacturers or different sources will make products with slightly different activity, and they are quantitated not simply by mass, because maybe there's some, say, extra protein when you are doing the mass of your protein with just, some general way of looking at protein concentration. But you actually look at the activity of the protein. And so you're looking to see, how much activity does this have? And again, I think about this with cytokines. And so usually it's something like how much of this cytokine is needed to make 1 million cells proliferate.
Yeah.
Or something like that. And that's defined as a unit. And the idea is that different manufacturers will have sort of different purification protocols and things like that. And so we tend to use this activity-based measure instead of a microgram-based measure. And usually if you really, I don't know if I can emphasize the reallys enough, dig, you can find the conversion chart.
Yeah.
But the conversion chart for my product is specific for my product in terms of converting between units and micrograms. And it may not necessarily apply for your product.
Yeah.
And I agree, Mark. I actually find this all very frustrating.
So AU would be activity units or
Maybe.
something like that?
Something like that, yeah.
And so what you need is, if for any given product, a definition of what an activity unit is.
Yeah.
And you're saying there's no industry standard.
No, there is no industry standard. And so if you look, like I said, if you look carefully or really, really deeply, you can somewhere find their definition of this many AU is equal to this many micrograms. But that is not easily findable in most cases.
So it reminds me of basically any kind of enzyme activity. You think about restriction enzymes, and you have to define what a unit is, for a restriction enzyme, or remembering in the old days reading biochemistry papers where they did purifications and they started with huge numbers, huge amounts of material and not much specific activity. And then it got down to less and less material, but higher and higher specific activity. So defining the units for your assay is important. So yeah, having that conversion would be good.
But I guess the point that I'll make to Mark is so, sometimes I will report some data from my lab where we are looking at cytokine measurements, and I'll say, well, we got this many units per mil of cytokine. And I'll talk to a non-immunologist, and they will want the micrograms. And the problem is that I was using a standard and a kit that gives me an answer in units. So I actually have to then go back and find, well, where did the kit get the standard from, and what was the manufacturer? And so sometimes it's pretty difficult to kind of make all of those leaps.
Okay, one more. Rich, I just moved one up from Monique there.
Cool. You want me to do that one?
Yes, please.
So this is relevant to a discussion we had on the last episode about vaccine manufacturing in Canada. And I think the comment was that there was none. Canada's vaccine plant history, this is Mark writing. Canada's vaccine plant history is basically, we had two major plants. See, for example, history of Connaught Labs. Connaught was nationalized, then privatized, and ultimately sold to foreign ownership. Both owners, GlaxoSmithKline and Sanofi, downsized the Canadian facilities during corporate takeover. Canada was ultimately left without vaccine manufacturing capacity because concentrating manufacturing at overseas plants was deemed more streamlined. Canada has now announced millions to build a new plant. All this is detailed in the article below. It gives a link at the end of the email. Vincent, you're right to be upset with the idea that Canada wouldn't be investing in vaccine manufacturing after all this. Good thing it's not true. Hope this is good news and makes you less grumpy. You haven't been so grumpy lately. Just a joke. Cheers. Keep up the great work. Loved hearing about TLR2 since I work on NF-kappa B signaling. Fascinating stuff. Cheers, Mark. Well, good.
Yeah, we had a bunch of emails, outraged emails about this. Well, the fact is they're stuck right now, and that's what the outrage is about. Yeah. I mean, yeah, they're going to ramp up, but it's going to take 7 years to them to get, and that's fine. But a lot of people wrote and said, you guys should do your research before you say anything. And that's why I said up front, above, it just came up.
Yeah.
So anyway, the fact is they're stuck. Canada's stuck. They're dependent on other countries, and that's the lamentable part. I think it's great that they're going to get back into it. Wonderful. Mm-hmm. It's really good. All right,
well, it's happened with other kinds of things too. Outsourcing of various kinds of manufacturing and stuff that, hasn't been happening in the United States. And so then we're at the mercy of getting it from somewhere else.
Oh, we're not free of guilt either. Yeah. I mean, we manufacture stuff elsewhere and now we're stuck. Yeah.
So will we learn from any of this?
No, economics drives everything, right?
And can we have global cooperation to try and monitor the situation and get on top of future outbreaks? I don't know.
Yeah, that reminds me of an interview I heard with Larry Brilliant this morning on Marketplace, talking about how we need to plan better and have a global plan. I meant to find the link and send you guys earlier. I'll do that.
I once invited Larry Brilliant to I think it was a TWiV at ASM meeting, American Society for Microbiology. Or no, it was ICAC. Either way, it's like, you have to plan 10 months ahead of time. And he said, I can't plan that far ahead for anything.
Well, maybe we can get him on a TWiV.
Wow.
Obviously, we don't plan anything.
Apparently not. Yeah, apparently not.
Actually, I know a guy who knows Larry Brilliant, so I could work on that.
I mean, he said, I'd like to do it in principle, but I can't plan 10 months ahead of time. I mean, give me
how about
break.
A week?
You ever hear calendars? Well, I think it was just an excuse. So Mark said I haven't been grumpy lately. It's true. Yeah. Yeah. I haven't been grumpy, although not on TWiV anyway. I'm grumpy elsewhere. Very grumpy elsewhere. All right, let's do some picks of the week. Brianne, what do you have for us?
I have a slightly sad story, which is about a rock formation in the Galapagos called Darwin's Arch, which collapsed due to natural erosion recently. And I was sort of really struck by this story and sort of the idea of, making sure that we value what we have when we have it. Because I think that, the photos of this are beautiful and I wish I could have seen it in person, but, so we should, take advantage of what we have while we have it.
Mm-hmm.
Very nice. I like that.
Kathy, what do you have for us?
I picked something that Justin sent me a while back, the woman who discovered the first coronavirus. And so this is a woman who had little formal education. She was born in Glasgow in 1930. She was a lab tech, and she got married and moved to Toronto. And but when she was in Glasgow, she was learning how to do visualization of viruses, or maybe, I guess she developed her skills more, at least with the electron microscope, when she was in Toronto, Canada. And then they wooed her back to London. And she was collaborating with Dr. David Terrell, who was running the research at the Common Cold Unit at Salisbury. And so they looked at this sample from a nasal wash, and they knew it could transmit common cold symptoms to volunteers, and they could grow it in organ culture, but not in cell culture. So they sent her the samples, and I haven't said her name yet. Sorry, June Almeida. And she saw the virus particles in the specimens, and she described them as like influenza viruses, but not exactly the same. And she had actually seen particles like this before while investigating mouse hepatitis and infectious bronchitis of chickens. So those of you that are thinking, those are coronaviruses also. Anyway, her paper to a peer-reviewed journal was rejected because the referees said the images she produced were just bad pictures of influenza virus particles. And 2 years later, she got it published in Journal of General Virology. And according to this article, Dr. Terrell, Dr. Almeida, and Professor Tony Watterson were the ones who named it coronavirus because of what it looked like in her images. And she eventually was awarded a doctorate from postgraduate medical school in London. So, a woman took the first pictures of coronaviruses, and I just thought that was cool. June Almeida.
I know the name. Yes, I've heard it before.
Mm-hmm.
I'm looking
Excellent.
up when the first coronas were identified, maybe not looked at, or maybe it wasn't a human. I think it's 1937. Is that possible?
I don't know. They said that this discovery of this nasal wash was written up in British Medical Journal in 1965, and then her photographs were published.
Hers was a human one, yeah. But
Right.
I think animal coronas were identified, but not looked at in 1937. Here you go. Cultured for the first time in 1937. Infectious bronchitis virus. Yeah, the other one she looked at. Yep, cool.
Yep.
That's neat.
Mm-hmm.
So she probably... Do you think she was the first to look at any corona under the EM?
Could well be.
Maybe.
Could be.
The iconic pictures. Cool. Rich, what do you have for us?
So I may have picked this sometime in the distant past, but it's time to do it again. This is an interview that was produced for the long-running BBC documentary program called Horizon, which focuses on science and philosophy, and it's an interview with Richard Feynman, who was a Nobel Prize-winning physicist who kind of marched to the beat of his own drummer. And it is a delightful interview with great insights into the workings of a free-thinking scientist's mind. I recommend it to anybody. It's the deal. It's the site that I found it on peppers it with these 10-second commercials, which are really annoying. But I think you have to subscribe to BBC Horizon to get it from their site. So it's behind a paywall, the original. But this is a This is a copy that's, if you can put up with the commercials, it's well worth your time. It's about 50 minutes long.
Cool.
Yeah, your quote was great.
I love this guy. And if you like this, he published a couple of books that are crazy that we've picked before.
Yeah.
All right. He's a free thinker.
And he's got those lectures too that you can see. They're great. Yeah. Wonderful stuff. My pick is a project called the Corona Project. Corona stands for COVID-19 Registry of Off-Label and New Agents Projects. It's a project run out of University of Pennsylvania by David Feigenbaum, where he tracks all the clinical trials, the COVID clinical trials that are looking at drugs, repurposed and off-label, that sort of thing. And you can go, there's a database, you can go search it, and you can go look for ivermectin, and then they give you a summary of whether the outcome was favorable or not favorable as well. So you don't, you could get a first glance of what's going on and see what's happening. It's really cool. I learned of this on a phone call recently, and thought it was pretty neat. And he basically said recently, okay, here's all the ivermectin studies, and none of them are really definitive. About half of them say there's no effect, half of them say there's effect. What we need is a 4,000 person randomized clinical trial to really get an answer, which I thought, who's going to do that at this point? Well, maybe somebody will. Anyway, and I invited him onto TWiV, and he's coming on in a couple of weeks, so he can talk about this. But cool. I think it's pretty neat, and he can talk about some of the findings as well. So, Corona Project. I think you go down to the data viewer, and then you can search it, and let's do ivermectin. Ivermectin alone. There's 62 trials underway, and one trial has been completed. So you could click on it, and you go view the outcomes, and then it gives you a color chart, overall outcome, negative, undetermined, unknown, undetermined, negative, mixed, positive. There's one positive for ivermectin. Anyway, that's why it's cool. You could get a glance. He said, if you don't want to read all the papers, which is really hard to do for some of these, you can look at that. Or we can ask him about it in June. We have a couple of listener picks from JH. Writes, at least with COVID, some people seem to recover completely. In contrast, if you're a cicada and with Massospora, and he sends an article in the South Bend Tribune, fungus full of psychedelic drugs could cause Indiana brood X cicadas' butts to fall off.
Wow.
There you go. A fungus laced with the same chemical as psychedelic mushrooms invades their bodies and eats away their insides until their abdomens crack, fall off, and get replaced with a ball of white spores. Wow. Nature's tough, so that's the spores, Massospora.
We have this phrase that I share with my grandchildren as we watch nature shows, that nature is a horror show.
Yeah, it is. Indeed. And we have one from Alan. I'm a listener from Dublin, Ireland. My own background is in computer network engineering, but I'm basically a nerd of a different color. A little over a year ago, I realized I didn't know what mRNA is, and so spent this past years delving deeply into molecular biology and virology. To say that it has blown my tiny mind is an understatement. As part of my nerding on these topics, I came across your podcast, have been hooked ever since. While the discussions often go over my head, they serve as pointers to topics to subsequently explore more deeply. It's been a trip. Anyway, among my other interests that overlap with frequent picks of the week are all things space-related. Recently, you have marveled over various SpaceX feats, so I thought I'd draw your attention to this book, Liftoff, Elon Musk and the Desperate Early Days That Launched SpaceX. The book covers the early years of SpaceX from founding through to their first successful orbital flight. It's a heck of a story and a great read. I'm certain you will enjoy. Thank you for all you have done over all these years, but especially in this past year in sharing your knowledge and experience with the rest of us. You have done a great service, Alan. That's cool. Maybe you want to read that, Rich.
I'm just bookmarking it here, yeah.
Okie dokie. Thank you for your picks. That'll do it for TWiV 757. Oh, by the way, there's one email here. Let me pull this up and read it. This is from David. He says, she's an MD, PhD, okay? And I'm saying that for a reason. David writes, periodically someone mentions, put it in the show notes, which sound very interesting, but I cannot find them. Are the show notes private to the speakers or available to all? For God's sakes, at the end of every episode, I say, that's TWiV 757. If you'd like to see the show notes, go to microbe.tv/TWiV. So that tells me, David, you never listened to the end.
Oh, man.
As probably 99.9% of people do. No, that's fine. It doesn't matter. But, you know, in a class when you're teaching and you say, here's how it works. And then a second later, someone says, how does that work, by the way? Doesn't that get you that haven't been listening? Anyway, the show notes are at microbe.tv/TWiV. And it's all the references we put in. So you can check them out there. They're open to everyone. I slave over them twice a week, 3 times a week.
Yeah, and that we get them. I haven't looked at one lately, but they get time-stamped by Jolene.
That's right, Jolene.
So you can,
oh, yes. Yeah. Thank you, Jolene, for doing the timestamps. Yes. It's very kind. She goes through and listens and then puts a time for every reference. And
you can find all the letters.
Find all the letters there as well. And, you know, there's 757 episodes of show notes all there for your perusal. And probably, I think some people do look at them. I think a lot of people, that's where you can find the pics, right? And you could
find the links to the pics. Yep.
So check them out at microbe.tv/TWiV. I should probably say it at the top of the episode. I know that, but I always forget. Anyway, if you want to send a question or comment like David did, where do I find the pics? At least he got that, right? TWiV at microbe.tv. If you like what we do, consider supporting us. microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thank you, Brianne.
Thanks. It was great to be here.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. I enjoy hanging out with you guys.
You do it a lot. I got to say, we spend a lot of time together.
Great stuff.
Especially in the past year. Oh, boy, it's been a lot. I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.